Good afternoon, everybody. I came in late last night and today's been a whirlwind. And with Joe not being able to present, I think I'm going to do double duty and try to cover both an overview of what we've been doing at the Jackson Lab for a number of years now related to peroxisome dysfunction, and then expand into our newly funded R24 grant from NIH to really build a mouse peroxisome resource. So a couple of things, and I noticed this on my badge just a few minutes ago, was Jackson Laboratories is an antibody company based somewhere else, so don't look for me under Jackson Laboratories. There's also Jack's Brewery in New Orleans, so don't look for me there either, except in Louisiana. The Jackson Laboratory is a legacy institute based in Bahamas, so we're frankly not even connected to the mainland. We're out there on our own little island, and the idea is to kind of create a scientific nexus where we can live in relative isolation and just think of all the great things that we can accomplish. But the world has caught up with us, and I think that with the advent of CRISPR-Cas9 technology, in 2015, we really jumped into rare disease research in a big way. But just to give you an overview about the lab, this is about a year old, and we're already, these numbers are way underestimated. So we've got about 2,000 employees now, and actually significantly more than that if you include subsidiaries that we now have in Japan and China. The idea is that we want to generate mouse models, but we also want to distribute them. So most of our Japanese and Chinese subsidiaries are involved in animal distribution, and Connecticut is our human clinical arm. So we have Jackson Lab scientists that work in Connecticut and Farmington that are really trying to cross the translational boundary as well as take on clinical disorders that may have preclinical sort of areas that we really want to get into. California services a lot of the pharmaceutical industry out there on the West Coast. In Maine, we're at the Legacy Institute. We tend to be mostly preclinical. We're focused primarily on developing mouse models that are applicable to human disease. Plethora of scientists, MDs, DVMs, and we cover all the research areas. And when I went to the lab to postdoc way back in the 1990s, I really wanted to get into mouse genetics and mouse genomics, and I figured why not go somewhere where they did only everything on the mouse, nothing else. So I kind of missed the software and yeast to some extent, but it is good to have neighbors down the corridor that you can just knock on their door and instantly you're immersed in sex determination or diabetes or cancer or neurodevelopmental biology. The lab is also a big distributor of mice, so we're a non-profit institution, and the revenue which is generated from the distribution of mice is fed back into the non-profit research arm. So that gives us a lot of capabilities to be able to throw money at projects in a way that many other institutions cannot. And that's really what triggered a lot of our early animal models on the peroxisome area. And we've been working for quite a while now with various foundations. These are patient parent foundations dealing with rare disease. We've also tried to reach out to industry to establish relationships so that we can actually move our models forward from the natural history phase of the analysis of the models into the sort of therapeutic area using AAV9 gene rescue, base editing, prime editing, essentially any kind of rescue approach that we might be able to try. And our in vivo pharmacology teams operate as a fee-for-service organization to work with the pharmaceutical industry to actually test new drugs on the existing animal models. And this is just a small subset of the groups that we've been working with, and I'll be glad to update this with the GFPD very shortly. So I already touched on CRISPR-Cas9. I'm sure it's not a new technology to many of you, but essentially what it allows us to do in mouse models is really for the first time to be able to mimic the clinical disorders. And we start from a premise, and that premise is that a mouse that is deficient in a gene is not the same as a mouse that has a missense allele in that same gene. And we've seen this time and time again. Missense alleles can be dominant, they can actually have hypomorphic characteristics, they couldn't be nulls, and many clinical alleles are nulls. But the classic study in mouse communities historically has always been, well, you take a mouse with the most severe phenotype and you study it, and that tells you what the gene does. Well, actually, what you learn is how the animal adapts to the absence of the gene, not really what that gene does. And so CRISPR-Cas9 really allows us to interrogate that. And not only that, but we can think about what mouse strain is most appropriate for that study. For those non-mouse people amongst you, you can think of mouse strains like different breeds of dogs or cats. They're all dogs collectively, or they're all cats, but each one of them has unique characteristics: their size, their shape, how fast they run, how obedient they are, and cats are never obedient. But basically, how they behave, and more importantly, how they respond to different diseases. And so there are some strains of dogs, cats that are more susceptible to various ailments than others, and it's the same in mice as well. And often that's a question that really we don't know the answer to. Our standard mouse strain is the standard black 6J mouse, but it may not necessarily be the right mouse under all conditions. But often we make it, we try to learn from it, and then we see if other strains can help. And I mention this because it's clear that peroxisome dysfunction is dependent upon the background genome. And we can prove that in mouse models. So you can also do conditional alleles using CRE-LOX-P. You can also create various combinations of alleles so the mutation is expressed during development, and then it switches to wild type after the mice are born or at a specified time. That's sometimes important because we want to know what the effectiveness of gene therapy might be. Not every dysfunction that occurs during embryogenesis can be corrected for. And it's important to kind of try to model that to some extent so that we can predict what is the best case outcome of a therapeutic intervention. And we've been playing around with methods of generating repeat expansions. So there are many diseases that are related to expansion of repeats in the genome in specific areas of genes. And we've been trying to model that. These repeat expansions tend to be incredibly unstable. So our pipeline that was developed over the last 7 years is actually fairly straightforward. It starts with essentially an assessment of whether or not we need a model development phase at all. Many mutants already exist throughout the world in various repositories. And so we start off by doing a thorough screen of what is available, and to determine whether it's actually quicker to get that mouse strain into the lab and develop it and then distribute it as a unique genetic resource, or whether we have to make it from scratch. If we decide to make it, we have various options. We can use zygotes, we can use venous cells, or we can use classical transgenic methodologies. And having then generated the mutation, we go through a really important phase, and this validation, I can't emphasize deeply enough how important that is, because you make a mutation with kind of a preconceived notion of what it's likely to do. The mouse may have other ideas, and so it's important to understand what the effect of that mutation is. As I said, a missense mutation could be a null, so it may be indistinguishable from another mutation which already exists, or it produces a stable protein, in which case you have to kind of understand whether that protein is going to interfere with normal biological function, or just kind of go off and sequester itself in a nice quiet area of the cell and not interfere. This is where we also look at the protein, because looking at mRNA alone often doesn't give you a complete picture, so you have to be able to assess what the effect of a particular clinical variant would be on the expression of the protein and its post-translational modifications. And that takes us to a point where now we've got a model we have validated to some extent. We understand genetically it's what it was intended to be. We reduce the likelihood of creating additional mutations elsewhere in the genome through our breeding program. And so now we're in the preclinical phase where we're really looking at its natural history. And we want to compare the phenotypes that we get in the animal with the clinical phenotypes. This is a really critical area, and this is where we work hand in hand with clinical researchers, Because what we want to do is to have them document, and as I see has already been done, a series of phenotypes which are associated with particular disease alleles. Now, depending upon the mouse strain that we've chosen, we're either going to have all of those alleles or a subset of those alleles, but we can quantify them, we can analyze them, and we can determine their reproducibility. And that allows us to establish biomarkers. And then we're ready to move to the distribution phase. So I'm glad to say that all of the mouse models that we make of JAKs are distributed license-free, and typically to any employee that requests them. We distribute globally. And we also try to build foundation relationships and collaborative relationships, because even though collectively within the JAKs lab we have a lot of expertise on different disease areas, we're not expert on everything. And so making a mouse model available, if we think it's going to help our disease relevance and discovery phase, we can initiate collaboration. So we do invite collaborations on all aspects of our models that we're generating. The boxes that you see are all different core facilities that we have available that we can dip into if we want any help in terms of developing a model or validating or understanding the disease relevance that it might have. So you can take that disease model pipeline and really kind of look at it in a different way. There's a common element to each of these pipelines, and that's going to be in the mouse breeding phase. So when folks call us up and say, we want to make this mutant mouse, and we agree to make it, we think it's going to be scientifically valuable, and they say, how long is it going to take? And we say, about 9 months to a year and a half. They end up being very disappointed. It's important to realize that most of that time is actually mouse breeding, and we can't actually accelerate that. So you have multiple ways into a development pipeline. You can look in ESL modification, or you can do a straight CRISPR-based knock-in allele, an exon loss, or a conditional allele. And they're all going to generate founders. Those have to be generated to N1 progeny. And then these N1 progeny are then further expanded to generate N2 progeny. This is by repeated back-crossing to the same red string that was generated on. And then we can generate sufficient mice to go to a head-by-head analysis. This is the first time we're going to generate homozygous mutants. And that two-stage process is really important. And part of our best practices that we've learned over the years is that we can't study mice at N1. We can't start generating homozygous mice there because there are off-target effects associated with CRISPR-Cas9. And we can't screen for off-targets in every case at the N1 progeny. We have to wait till we get to the N2 progeny. And that's where we see a discordance between the oligo donor that was used in the targeting experiment with the actual gene itself. And we have to see co-segregation. If we don't, it means that our oligo has also inserted elsewhere into the genome. It was considered for quite a while, but that really wasn't possible. Oligos are really small, but we have seen it on several occasions. That's why we always go to N2. It adds another 2 to 3 months to the timeline. And then we get to the evaluation phase and the distribution. One thing which will come up again is that we do a test mating of all of our flux alleles with SOX-T3, and I'll touch on that in a few minutes. So this is just looking from 2015 to 2020. Using that approach and using a kind of a semi-high throughput pipeline, we generated about 187 new alleles here. Now, they're not 187 different rare disease genes, because some of them were multiple insertions into the same gene as part of the same project. But essentially it kind of gives you an idea of how 3 people working relatively easily on a pipeline approach can actually generate an incredible number of mutations in a short period of time. And that leads me into what we have available at JAKs in terms of peroxisomal resources. So it starts out with the gene PEX1 at the top, and I've indicated its genomic structure here with the introns and exons. And using an allele that was generated by Stephen Steinberg at Hopkins, and that original donation at the top of the PEX1 TM1.2 SJMS allele, this then developed, from that resource we were then able to develop additional resources. So when that was donated to the lab, it was on a mixed segregating background. It was a B6N and a 129 background. And primarily through work of Nancy, who indicated that there was this concordance in the phenotype depending upon the genetic background. And the B6 and 129 were the two players here. So we ended up generating from the original stock strain that was delivered, the repository and Nancy independently developed two lines. So Nancy generated the same mutation, moved on to the 129 background, and we generated that mutation on the B6 background. And those are now also available. On the left-hand column here, you can see we've got JR numbers and MMRC numbers. There are two different mouse resources at the lab. As a customer, it's pretty much invisible to you, but there are fundamental differences in how they operate. So the MMRC is a federally funded program to make available mouse mutants that have been published on. So they have to accept them. The Jackson Lab repository is internally supported, and it takes on strains that it thinks are going to be of general use to the scientific community. And it's possible that the internal repository can reject a certain strain from being accepted into the program. And many people, when they hear that, they say, Jack sucks, I can't get my mouse there, I really wanted to distribute it. The answer is, go via the MMRC. It may be deposited in JAX, and may be deposited at any one of the other MMRC centers in the country, but it will still be made publicly available. So going back to the PEX story, we now have PEX on 129, S6, and PEX on B6. And the allele that you can see up top is a little bit complex, so it's floxed for 2 exons, and it also has the G844D mutation, which is, as Nancy's already mentioned, clinically equivalent to the G843D allele. And so to simplify that, what I did was essentially rescue the mutation using another CRISPR-Cas9 targeting. So taking the mouse that was originally mutant and retargeting it to get rid of the 844D. And that creates a straight flox allele in PEX1. And so the original rescues were done in the B6 and the 129 versions, and so they would be flux for exons 12 and 13. And then I also separately came in and fluxed exon 13 by itself, and also created a new null allele in PEX1, which is deleted for exon 13, but doesn't have any LOXP sites in it. And the reason for that is here. So the B6 and the 129 alleles for this PEX1 mutation, the G844D allele, has a phenotype which is dependent on the genetic background, but it's not just a straight difference on the background, it's actually a combinational effect. So on either one of those two backgrounds, on B6 or 129, those mice, they're born small, and we see lethality around about 15 to 20 days of age. It's about the time, and I was listening to the metabolic sort of discussion going on there, it's about the time when mice are transitioning from suckling a high-fat milk composition from the dam, and they're transitioning to generally the high-carbohydrate chow diet. So one wonders, based on that, whether nutritional aspects of these models could actually be explored in a mouse model more thoroughly. What we did find, though, and Nancy's original observation on her background as well, that led us to say that we could actually generate mice on an F1 background that are essentially 100% viable. They're still small, so we haven't rescued the size of the animal, but we have rescued the viability. This is a much better experimental model because we can consistently generate and regenerate homozygous mutant mice to evaluate for therapeutic evaluations. Now, there are survivors that come through on the individual strains, but they're rare. But they do exist. But because you can't actually get a lot of them in an experiment, your experiment either has to be huge, or you just have to accept yourself to a rolling star mechanism. The lab approach, where you buy 2 different strains and then just cross them together, is a much effective way to generate homozygous mutant mice. The issue of the flox allele came up with the results from this data. And I promise you, this is the only data I'm going to show. A lot of it is overview. And it came about through some observations where we wanted to determine how well these LOX-P sites work. It's a standard part of our model development pipeline that having inserted LOX-P sites, even though you can actually see them by sequencing, we need to confirm that they're functional. And our tool for using that is the SOX2 Cre driver. SOX2 is expressed maternally, and all embryos see it, regardless of whether or not they have the transgene or not. And that way, it's a very quick and easy way of determining whether your LOX-P sites are functional. It's also a very strong driver, because if it gives you problems here, you may have problems elsewhere. And one of the things I want you to kind of, and I can summarize all of this for you, is that the exons 12 and 13, they're about 2.5 kb, 2,600 kb apart, which is a little big for when you're thinking about an effective pre-deletion strategy. And that's what we see here in the mice that have the knockout allele or the conditional allele, right? It's 50% of the mice that are coming out of this. So this 61 to 31, 21 to 11, that's completely expected. But of those 31 mice that we knew carried the variant allele, only 10 of them had a clean knockout and no unwanted flox allele present. So there was always a background level of the original unfloxed allele in a mouse which shouldn't show any evidence of a floxed allele at all. So that ends up being about 32% or so. When we looked at that in the exon 3, it still occurred. About half of the mice, 54%, still had evidence of a non-targeted allele in the presence of the Cre driver. But differently, when you look at mice that actually had Cre, so within that subset of mice that are Cre positive and they're Cre negative, you can see that even in Cre positive mice, we still had some mice that still showed continued evidence of the flox allele being present, so a degree of mosaicism, if you like. But none of them that are Cre positive on a smaller deletion, which is only 600 nucleotides or so, showed that evidence. So it tells us, and this is not a rigorous study, but I think it kind of, we raised our eyebrows a little bit at this data because it's atypical. And it tells us that this region of PEX1 is not actually that accessible early in zygotes. And so Cre doesn't really see it very well. It may be part of inactive chromatin. Or it tells us that we need to be very careful in evaluating the results of experiments where Cre is used to turn off PEX1. You have to assess the possibility that there may be cells still around that have not been exposed to Cre, and that you may end up generating a chimeric animal with both null and wild type alleles present. And this may suggest that we need to make an even better one. So what resources does the lab have that are not PEX1 related? Well, we've actually been looking at several other genes as well. PEX5, PEX10, PEX7, PEX16, and we also generated the ABCD1 knockout to the excellent history model. And they were generated in either through Joe's insistence or through collaborations with researchers. And I know a lot of our PEX10 work was done with Andrew Lonecker, who has a son who has the H288D allele. And our goal here is always to generate them, to characterize the models, to understand what might be happening. In the case of the PEX10 H288D homozygous mice, they're small and relatively inactive. We have to euthanize them pretty quickly because they really don't thrive at all. But loss of PEX10 is homozygous lethal. So, we do see some evidence that H288 is hypermorphic, and there must be some residual expression that rescues the animals to get them to a point where they're actually born. Because of that, issues with lethality, we're generating floxed alleles, And we're doing that by taking the allele that was generated as part of the COMP project. So the COMP project was a global initiative designed to try to target every gene in the genome. And although they decided not to do that, they did target a significant number of them. A lot of their first generation models were just straight knockouts, but their second and third generation models were more complex, and they include flux alleles. So the way that it works is that the 1A allele is a COMP allele. So JAKs cannot distribute that. If you want the 1A allele, which is sometimes but not always hypermorphic, it could be a null, it could be a hypermorph. You have to go to the European website, you have to go to UC Davis, where a lot of these COMP strains are captured and held in repositories. But we are able to take a 1A allele and convert it into a straight flux allele, that would be the 1C allele, and JAKs can distribute that. So that's what we have here. We generally bring in mice from either the European Mouse Mutant Archive or from UC Davis, wherever these mice happen to be. We then essentially eliminate the LAC-Z and NEO, which forms the 1A allele, to make a straight flux allele, and then we test it, and then we put it out onto the website. So we can learn a lot from COMP, because as well as making mouse mutants, they also phenotype mouse mutants. Our rare disease program at JAKS, basically we were able to scrape together funding just to generate the models. And we were hoping that our collaborators or the scientific community, once these mice were available, would then go in and fill in the gaps as to really what was the underlying biology of this mouse and what could be learned from it, other than what we were doing was a very basic one-pass analysis. So for the PEX1 mutants, now they fluxed exon 8 the gene, but they find against homozygous lethal as we do. This is all on the B6N background. But they did see a cardiac phenotype that we've not interrogated on some of our own models. And they see the abnormal liver that we see as well. We see very fatty liver in these mice. PEX3, they generated the TM1A allele flanking exon 4, which actually seems to behave either as a sort of hypermorph, but it could also be a null. And they see an array of different phenotypes, primarily metabolism being an endpoint, but they also have eye disease. That decreased body fat could be tied into those metabolic aspects as well. HEX6, 11G, 14, there are 26, so null alleles are all being characterized, and they've come up with an array of phenotypes. Now, they do this as part of a relatively high throughput Shepherd's screen, so all of this data has to be revalidated within an individual lab. But it does represent a good place to go, and mousephenotype.org is generally where you can learn, do they have this allele? Do they have mice? Have they done phenotyping? And some of that work is still pending, like the PEX16 analysis. The interesting thing with that one is that they see a difference in body length, which is only applicable to the males, but not the females. PEX16 doesn't map on the X chromosome. So with that, I'll finish up the JAKs portion of the talk with relationship to the mouse models, and then I can switch to Joe's part of the talk. I'm going to use Joe's original slides, excuse me, but I'm not going to replace his style. So you're going to have to bear with the English accent for a little while longer. But for those of you that don't know, this is Bar Harbor. This is the Jackson Lab in Bar Harbor next to Acadia National Park. So I think my lab is somewhere buried in there. Usually it's actually in the middle. And the mountains behind is that Champlain Mountain view ahead, and that's the Gulf of Maine up above. And it's great when we have hurricanes moving up the East Coast because it gets very interesting to be out by the water. And of course, making mouse model takes a village. And Kathy Lutz started a lot of this work on the peroxisomes in terms of getting the funding together to make the models. And then I took over, and my team was also represented there. And of course, a lot of this was driven by Joe and Nancy in terms of providing input, ideas, and suggesting what would be a good model to think about. Hey, are there any questions first on the JAKs component, how we work and what we do?
I just had a question on those compounds, actually. I don't understand how they
So I don't have a slide, unfortunately, of the actual graphics, but they have that on the COP website. So the 1A allele contains a promoterless LAC-Z with a neo-selection. So all of these models were generated by ES cell manipulation. And there's a splice acceptor in front of the LAC-Z gene. So the idea is that when this cassette went in, the splicing would be perturbed because now LAC-Z was being expressed, and therefore you could use LAC-Z expression indicator when and where the promoter was active. But it was a very weak splice acceptor that they used. And so in some cases, you can actually completely bypass that, and then you have normal splicing into the downstream exon. Now, downstream of the LAC-Z and NEO is LOXP sites flanking a critical exon. And there are Fritz sequences that are flanking the LAC-Z and the NEO. So you can make the 1A mouse, which carries LAC-Z, NEO, and 2 LOXP sites, and make it with flip, and get rid of LAC-Z and NEO. You get rid of the splice acceptor, and now you should have normal splicing in, and it should just be a straight flox allele. That's the 1C allele. And you can also just do a LOXP mating to start with, and then you end up with LAC-Z and NEO being expressed on another background. That would be the 1B allele. Or you can delete everything, and that would be the 1D allele. So there are 4 alleles that can be derived from 1A. The point of 1A is that you can use LAC-Z expression to determine when the promoter is active, but it's possible that the mouse may be a hypermorph or it may be a null. You don't know until you test it.
Some of those phenotypes is bizarre, that you pull down on that slide.
And it's a high throughput approach, but they generally have enough animals in there to actually have some reasonable good estimates of what might be going. What is missing is you don't really get a good handle of when they assay these animals. Was it 1 cohort? Was it 2 or 3 cohorts? That's why they basically end up being guideposts that you can use, saying if you're interested in retinal dysfunction, here are some mouse models where others have reported this, but you have to confirm it for yourself because they don't guarantee it. This came about because the first round of the mouse mutant analysis was the ENU screen that was done in the 90s. And so ENU mutagenesis was used to generate lots and lots of mutations scattered throughout the genome. And they mapped them, and they ended up marketing them as specific ENU mutations generating specific genes, but there was no phenotype data associated with them. So nobody wanted them because who has the money to buy a mouse, to phenotype it, and then to determine it's not what we wanted? You've now spent a lot of time and energy on that, which is why when the COMP project was developed as an initiative, it had to include an element of phenotyping. But of course, it's high throughput. It's not the same level of critical analysis that invested research would give that project. But it's enough to tell you what was, that they didn't see an eye phenotype means that they didn't see it on the high pass screen. It doesn't say that phenotype doesn't exist. It's just they didn't see it. So I don't know if it helps or hurts. In the end, I think it helps more than it hurts, but it's an imperfect solution to an issue that really still exists. People don't have money to validate a model every time they want to buy it. When they buy a mouse, they want to buy a mouse which actually will replicate the disease phenotype that they're interested in. So this is when I put on my Joe hat and to talk about the R24. So this was something that came about organically, I think, through the continued interest in the Rare Orphan Disease Center at Jackson Lab, my team's effort in making a lot of these models, can we collectively group them together and actually seek funding as a resource? And if we did that, what else did we want to do within that space? And Nancy and Joe, co-PIs with me on terms of the R24 application. We were successful in getting it funded the second time around. I think it kind of highlights the fact that the first time you're always going to get kicked in the teeth. But if you get up and actually kind of think about what it is that they really wanted, you end up passing it in language. I don't think our goals changed at all. I think it's just how we expressed ourselves was the major breakthrough. So the initial proposal, what that NIH was looking for, was that they said there was a funding opportunity to develop projects around certain areas, and that could be developing, characterizing, or improving animal models for human disease, improving access to data, generated from these animal models, improving diagnosis and control of diseases of lab animals. Well, we kind of X'd out 3, but focused on, hey, we could really make a difference with 1 or 2. And a critical element of this R24 mechanism was that the disease model had to span different interests of different NIH institutions, and peroxisome dysfunction clearly meets that criteria. So what they didn't want to function as a rare disease resource that was only specific to a particular disease area, but the argument we made, and is being made today as well, was that peroxisome dysfunction spans a number of other disease areas, and that should be leveraged, and then NIH should be aware of that fact, and that's what I think really bumped up our funding score. So out of that, we developed 3 specific aims, 2 of which are certain to be done at Jackson Lab 1 and 2. And aim 3, I think, is a little bit in the air right now. We originally written it as a Jackson Lab-centric endeavor, but I think, and I'll kind of go into a little bit more detail about where we are with the antibody resource. But this is a community-driven R24 mechanism. So we're really, it's an interaction between the Jackson Lab, and sort of USC, where Joe is, and McGill, to come up with some standardized approaches and methodologies, but to look for constant feedback from the scientific community and from parents and foundations, to try to allow us to develop the models that are going to be most useful to you. So here's my pitch. There's a free mouse that I'll give every one of you that you can kind of tell me why it's really important, and you can convince our internal advisory board that that's a direction you should go in. And we'll take care of making the mouse, and we'll give you some baseline data, but you got to do the work. And it would be really good if you did it in collaboration with us, because that would make us look good, but also make the company look good as well. Because it's a 4-year grant, but it's renewable 1 more time. So in 8 years, we'll be back with, really a lot of problems, because that resource will go away. But we want our renewal in 4 years to look as good as our original application, because then they're going to be saying how well was the resource used. So think about what your animal models are. What are you missing in the lab that you would really like to see in a live mouse? And we can get that to you in 9 months to 1.5 years. So specific aim 1 really takes advantage of what people have already done, but haven't kind of taken it to the next level. We want to make mouse models which have already been published upon, if the investigators and the institution where they reside are amenable, to make that available to the scientific community at large. It means that you could remove that from your colony if it's no longer an area that you work on, and you can just deposit into the peroxisome resource at Jackson Lab, and we'll take care of all of the distribution for you. Well, I say we, it'll be my colleagues working on the repository side. Specific aim 2 is to develop the mouse models that either researchers or their institutions refuse to make publicly available, but nevertheless have valuable components to them, or to make brand new models that nobody's ever made before. We'll do all the legwork in terms of trying to figure out whether somebody's made it, has buried it somewhere. But if it's a model which hasn't been made before and it has a clinical relevance and will further the aims of the R24, we'll go ahead and make it for you. And aim 3 is really a little bit more complex, because it deals with this whole issue that we've been addressing for some time now in the rare and orphan disease space, and is that a lot of these genes that are part of rare diseases are fundamentally important for basic processes, but the tools available to analyze them are much more limited. So companies are very happy to sell you antibodies. What they won't tell you is whether or not the antibody is any good. They'll swear up and down that their antibody is perfect and it's specific and they did it this way, but they lack basic information. Either they choose to withhold it or they just don't know, and they won't tell you, is whether or not it's actually specific for that protein of interest. And also, one thing to be very careful of is that an investigator, if they make an antibody and then they license it out, these companies will license it to each other, and then distribute exactly the same antibody under different names. And so traditionally the way is I'm going to buy an antibody from company X. Oh, it didn't work. All right, it didn't work. I'm going to go ahead and buy a different antibody from a different company that recognizes the same thing. How do you know you're not getting exactly the same antibody? So there's a crisis going on in terms of antibody validation. And we felt that we should at least try to make some inroads in that. And what can we offer that would be advantageous? Generally, we're looking for antibodies that would recognize mouse proteins. Added bonus, they recognize human as well. But we would also have available a mouse null lysate available, right? We made that gene knockout. Therefore, we have a way of validating the antibody. So the mouse validates the antibody validates the mouse. Both sides have to make sense. And that would be part of a routine screen. And I know Nancy and her group have access to a lot of cell lines from clinical samples where null liens exist also. And so we can test for sort of cross-species interactions with antibodies that are generated in order to confirm that they also see the same protein in the mouse. And if a lysate shows no expression of that protein, then it should not show up as positive. And to try to build that as a real resource, and we threw it in as an aim to show that we really were committed to thinking about making this information available, generating the resource, making it available, and telling people about it. So underlying all of this is a website that we're hoping to build around this resource, which will tell people, what mouse models we've been importing into the repository, when they're likely to be available, what mouse models we're planning on making, because we're not in the business of competing. If you have already started a mouse model and you see it come up on our website, then let us know and we'll start making it. Assuming that at one point you're going to make that available to the community at large. And the same with antibody resources. We really need your help here. What antibodies are you using? Where are you buying them from? And why do you think that they're good, or why do you think they're not good? And you can capture some of that information. You've got to deal with the lawyers on blasting companies. But we basically try to generate a set of tools that the community at large can agree upon that would be really save us a lot of time and money in chasing down these issues. So the R24 mechanism has an internal as well as an external advisory board. Miriam Bias, who's here, I believe, is the head of our external advisory board. Joe, I, and Nancy are also on it. Jean Bennett, Sami Posey, Yang Chime. These are technically experts that we built around this area that cover a range of expertise that would give us an appropriate. If your name's up there and you're surprised to see, I apologize. This is Joe's data. But essentially, this is so that we stay on the even keel. NIH is very vested in making sure that any sort of multi-center grant works under the auspices of the external advisory board. You guys have a lot of power on the external advisory board, and you can determine the direction and the scope of our work. And our internal advisory board basically is more mechanistic, how we're doing and where we're doing. There's Joe, myself, Kat Lutz, who's transitioned into running the Rare Disease Translational Sciences group. But she also brings in specific academic interest in ALS, Alzheimer's, SMA. And I think clearly we're in the process of uncovering lots and lots of relationships about the role of neurodegenerative models and the role of peroxisomes. And one piece of advice that I would give into foundations, and I must have worked with over 50 of them in the last couple of years, is take your rare disease and try to make it common. How do you do that? Is you try to connect it with a disease where there's a lot of money and there's a lot of vested interest in it. So connect it to cancer, connect it to some aspect of behavior which is prevalent. Autism, for example. But in this case, maybe we're in the process of thinking about a relationship between neuronal dysfunction that we see in ALS and Alzheimer's, SMA, Parkinson's, and the role of peroxisomes within that may be contributing to a subphenotype, but nevertheless a relationship that we can actually take advantage of and leverage to convince skeptics. Just how we're going to work with those mice that either come in or that either we make them, M1, M2, is that we would think about genetic standardization is number one. A lot of mouse strains that come in, everybody swears up and down, these are on B6J, they've never been on any other background, and then we go ahead and do sort of genetic analysis and we find bits of BALB somewhere and bits of 129. So the first thing we do is we're going to do a genetic screen, clean these mice up, because as we've already seen for PEX1, PEX7, PEX16, there may be underlying genetic variation regulating the severity of that phenotype. So it's important when we characterize these animals that we understand. We don't know where the variation comes from, at least today, but we need to at least make sure that what we're selling is as much genetically defined as it is phenotypically defined. We can look at gross behavior in terms of biometrics, we can look at expression profiling, we'll do baseline histology. Obviously, the tissues that we look at are going to be dependent to some extent on the peroxisome model we're looking at. But we can also do the metabolite profiling. Originally, it was put in to do with Kennedy Kruger, and Moshe's group was going to do a lot of these kind of analyses. Clearly, that's an area that we need to make sure it remains functional and truly useful to all of us. In terms of scope, I think that you can probably double these numbers. We were trying to be somewhat conservative with NIH, but our capabilities actually exceed this. So we can generate, we were thinking about 3 strains per year being imported. That is going to be what it is. Not a lot of institutions allow their mice to be donated into a repository without some kind of cashback as a result of sales. So that's something which has to be negotiated. But without our M2 multiple generated, we said up to 4 a year, we can probably go to 6 to 8 with no trouble at all. And M3, we've got to figure out how to best to generate antibodies, whether we use a mouse monoclonal service. Llama is a great way to generate antibodies now. They're single chain antibodies, and they're really nice to work with, or we can go with polyclonal potion rabbits as well. But that's a process under development, and hopefully we can resolve that very quickly on how exactly we're going to pipeline that. So, a couple of things that we are going to make. We're going to make a PTS1, a PTS2 reporter system. We're also going to create a log stuff blocks version of PEX1. I'm also looking to, my new responsibilities include developing new technologies to do better and better things in the mouse genome. And one of the challenges always has been transgenes. They've generally been cDNAs because they're the size limitation and how much new DNA you can exogenously add to the mouse. And random transgenesis has a lot of problems, as I'll be happy to go through with you after a couple of drinks in me. But we've developed a methodology now using BXB1 series integrates to move about 78 kilobases of DNA onto the genome in a single copy. That allows us to think about moving whole genes rather than cDNAs. And that's really important because one gene often makes more than one cDNA. We're also going to be looking at generating specifically patient-specific hypermorphic alleles. Again, with that criteria that a null mutation is not the same as a patient-derived mutation. So really we're looking for interesting patients that you have that represent a unique perspective on how the gene works. And we'll take that allele and we can generate it in the mouse. And look to generate, hopefully, an allelic series of different mutations of the same gene. And they'll all be on the same genetic background, so now we'll have a direct behavioral and biochemical analysis of each of those strains. And this is where we need your help in terms of getting information from you to us so that we can repackage it and send it back out to you. This is, again, one of Joe's slides, but and I think he's being somewhat analog here. I don't know anybody that uses Google Docs, spreadsheets anymore. So we're thinking about a website where we want to get information from you, collect it, and then turn it around and repackage it out in such a way that that information becomes useful to you and allows us also to do better validation studies on new mouse models. And your ideas on mouse models that you want us to make at JAX, these mouse models will be made available as a community resource, but we can ship you a couple of mice early so that you can tell us information that we need to know that we don't have the expertise to get some unique aspect of the biology that you think might be really interesting. And the hope is that we can develop that as a new collaborative relationship. And at that, and again, this is Joe's obviously excitement with Mickey. I'm sorry for taking so much of your time, and hopefully I got Joe's part in the mindset of where Joe was.